Thank you very much, and thank you all for being here. As was mentioned, I'd like to just share some preliminary data of a study that we began at the end of this past summer, in which we sought to assess the walnut twig beetle's ability to colonize treated black walnut wood. And once again, welcome to the epicenter of thousand cankers disease here in the native range for black walnut. As we're all acutely aware by now, the first confirmed instance of thousand cankers disease here in the east was made here in Knoxville. Since then, it has popped up in several other states, and as we are seeking to get an idea, a grasp on how thousand cankers disease is spreading, this will be one of the major issues we're faced with trying to manage with thousand cankers disease, is trying to manage that spread, with the ultimate goal being to try and prevent any further movement, or probably more realistically, trying to prevent any further human-assisted movement of the beetle and the pathogen, and then to mitigate losses in areas where thousand cankers disease has already been introduced and is likely soon to be introduced. One of the major tools that has already been implemented for several states are the quarantines, in which the movement of walnut wood is restricted, and phytosanitation requirements are put in place in order to render that wood safe to be removed from any quarantined area. A few other aspects in trying to manage for the spread of the disease are to identify any and all potential pathways in which the movement of the beetle and pathogen are facilitated, and to assess the efficacy of the regulations that we have in place. We need to be sure that those phytosanitation requirements are in fact working. To briefly touch on some industry issues, it's important as managers to both protect the natural resource, being the walnut trees, but also to keep in mind the industry perspective, as there is a major industry, the walnut lumber industry, built around this natural resource. A few major issues that we're aware of are the veneer. A veneer quality log is worth at least 2 times that of the lumber you would get out of that same log, and the restrictions in order to try and prevent movement of the beetle make selling veneer logs out of a quarantined area difficult for most mills, as the standard procedure is to leave the bark on in order to protect the surface of that log. Another issue that has been raised is the losses incurred from having to have squared edged lumber. You lose some of the wane, which is part of the rounded edge in a board that you cut from a log. And although from a regulatory stance, you're making sure all of the bark is removed to fully remove the threat of spread, but losing that sliver, those losses do add up. And then there's the treatment cost, another input cost. So these are just some things that we have to keep in mind as we do try and enforce these regulations and manage this spread. Some of the phytosanitation implements that have been adopted include heat treatment, which kind of including kiln-dried lumber in that the standard way of drying the lumber includes high heat temperatures, and fumigation, often with methyl bromide concentration. The squared edge green lumber that I mentioned previously. And so we've found several ways to effectively eradicate any infested wood. That kind of raised the question to us, well, once this wood has been treated, is there any further threat that walnut twig beetles can recolonize and get back into that wood resource thus reopening what we thought was a closed channel for potential spread. So in order to address that issue, in our study, we've implemented 5 treatments with 30 replicates per treatment. In our 5 treatments, the heat treatment, we took a clean walnut bolt and raised the core temperature of those bolts to 60 degrees Celsius for a duration of 60 minutes. The fumigated treatment was, again, one of the round bolts. They were placed into metal chambers and inoculated with a concentration of methyl bromide in order to meet the regulation standard. Then we had 2 lumber treatments in which we purchased lumber from a mill here in East Tennessee. We had 1 treatment with the bark left on, the other with bark removed, and this lumber was dried in a kiln here at UT to 8% moisture content following a standard walnut drying schedule. And our final treatment was another round bolt in which no treatment was added. That's our control. So each of these samples were given a walnut twig beetle lure and were hoisted into infested trees, where they were hung for a duration of 30 days from early August through early September of this year, and exposed to beetle pressure. The samples were all hung in a total of 30 trees over 11 sites throughout the greater Knoxville area. At the end of 30 days, the samples were brought in, cut in half, and either the top or bottom half for each sample was randomly assigned to our emergence barrels. Once that piece was assigned, we took each individual piece and thoroughly brushed that sample before placing it into the emergence buckets in order to remove any beetles that may have been lingering on the surface or in some bark crevices. The bolts were then containerized in our containers here with a funnel leading into a wet cup, and we are currently monitoring that emergence. We check the traps every 4 weeks and will continue to do so for 6 to 8 months. The remaining half, we took the middle third section of bark that was remaining and peeled that bark off, and we mapped the walnut twig beetle galleries using a map wheel in order to get an idea of the total gallery length. And in that section of bark that we removed, also tallied any walnut twig beetle life stages, including larvae, pupa, and adult. This was only done for the control, heat, and fumigated treatment, the 3 round bolt treatments. We realized that the lumber that we purchased was harvested from an infested county here in East Tennessee, so we couldn't say with confidence what was beetle activity prior to treatment versus post-treatment. Therefore, that wasn't included, and the bark-free piece, there would be no bark to assess anyways. So just to share some preliminary results, we have had 2 collections brought in and fully analyzed, so we have 2 months worth of emergence. And some things that we've seen or starting to see, a few things are not surprising. So just the mean total emergence numbers per treatment, the control bolts, as expected, have a much higher, significantly higher infestation or emergence rate versus our 4 treatments. The fumigated and heat treatment are not significantly different from one another, but are different from that of the kiln-dried lumber no bark and kiln-dried lumber with bark. And as expected, the lumber without any bark to date has not had any beetle emergence. Given the biology of this beetle, we wouldn't expect it to get into that wood. What was a little interesting is that we have found some emergence out of the kiln-dried lumber with the bark remaining on it. To break that down a little bit further, we had in the first 4 weeks of emergence, we had 11 samples that did produce emergence out of the 30 total. And they produced anywhere from just a single beetle to as many as 14. And one of the trends that is beginning to emerge in the data and has been holding true across our treatments is we seem to have somewhat of a site difference. The three most productive of these lumber with the bark on kind of shows that we have a few sites that have a heavy concentration of walnut twig beetle pressure, and then other sites where we just don't have that many beetles. And as I said, that is holding true across treatments. The second trend that I wanted to point out with this particular treatment is the drop-off that we see from the first 4 weeks to the second 4 weeks. We went from 11 samples that had emergence to just a single sample, bolt number 23, that had 2 further beetles collected. And this could potentially suggest that what we're showing is that the beetles can get into that wood and kind of assess and test for host suitability, but that they're not really staying in and utilizing that as a food resource, and they certainly do not seem to be reproducing within that resource. So to better compare our different treatments, we're really looking at the mean walnut twig beetle densities, so the total number of beetles over the available surface area of bark. The trends hold true as with the just total emergence, that the control is significantly greater than all the others, and that there is no significant difference between the fume and the heat treatment. For the mean gallery lengths, something that was interesting, again, the control treatment has a greater total mean gallery length, which would be expected given the greater pressure we found in the control bolts. But what was interesting is that the heat and the fumigated treatment, which in terms of density of walnut twig beetles were not significantly different, there was a difference found between the mean total gallery lengths. And this kind of just suggests that the walnut twig beetles are not likely evenly distributed within any given section of wood, and that we just didn't happen to pick up the same kind of density in our bark sample between those two treatments. And to kind of further support that claim, the life stage counts, again, what was surprising here is that the control treatment, the total life stage counts, was actually a little lower than you would have expected given the density of walnut twig beetles that were emerging out of those bolts, and that the only significant difference here exists between the control and the fumigation treatment, with overlaps between control and heat and the heat and fumigation treatments. That just further supports that the distribution within any given bolt is not even. The beetles searching any single point within the wood, you're not as likely necessarily to find the same kind of pressure. So some preliminary conclusions to draw here are that our treatments do significantly reduce the walnut twig beetle presence versus a no treatment. However, there does appear to be some level of risk, especially biologically so, I think, in the fumigation and the heat-treated bolts, that we certainly show there is still a biological significant number of walnut twig beetle recolonization. In our worst case scenario, in this study, we wanted to expose our treatments to as much beetle pressure as we could in order to see if is it at all possible for the beetles to get in and continue to utilize that resource after it has been treated, and that we have found some recovery from the kiln-dried lumber with bark on. This was certainly unexpected and is worth looking into further to see, to determine, are the beetles really using that resource, or is it just a potential hideout in which these beetles can still get into, which would, either way, would still leave the lumber with bark on as a potential pathway for further spread. So all of this to say, perhaps there is impetus for some kind of further protection in trying to keep the wood protected after it has been treated, in order to make sure that we have rendered that wood completely safe for transport out of any quarantined areas. And then just to leave you with a few final thoughts, as I said, this study, we exposed our treatments to as great of walnut twig beetle pressure as we could. I think the next step, now that we've found that yes, beetles can and will recolonize treated wood, the next step is to see, well, is there a mechanism of those beetles to find that resource, maybe given not quite such an artificial proximity to walnut twig beetle activity? Is there a mechanism for the beetles to find wood at a lumber yard, or about to be transported outside of a quarantined area? And then a final question is, so we've looked at the beetle, walnut twig beetle will recolonize. What about Geosmithia morbida? Will the pathogen, can it grow in the treated wood? Something that this study we just weren't looking at, which could be something interesting to look at in the future. With that, just would like to quickly acknowledge all of the help that we received in the field and in the lab with this study, and of course our funding sources, the Forest Service and APHIS. Work is not possible without them. And with that, I'd like to field any questions you may have.